All right, let's get started. Hello, guys. Welcome to our RNA-seq transcriptomics short talk session here at BIOC 2023. For those who haven't met me yet, I'm Wes Wilson. I'm a tumor immunologist and computational biologist at Penn Medicine, and I'm here to have the honor to chair this session with you guys. If you have any questions, we'll give everybody 1 minute of questions at the end after 9 minutes of talking. If they talk too long, then they may not get questions, and me and Jen will stand up near them in an uncomfortable fashion until they stop talking, sort of like huddled on one of those subway rides and people are just too close. If you have other questions online, people, please put it in the Q&A and hop in. We have moderators who will read your questions aloud to our speakers. And if you're here locally, you can put your questions in, hop in as well if you want to stay anonymous or you have a sore throat or any of those reasons. With that, we're going to jump right in because we are running a little behind with technical issues. So I'd like to introduce our first speaker, Noor Singh, who's a PhD student in computer science at the University of Maryland, giving a talk on tree-based differential testing using interferal replicate counts for RNA-seq.
Hi everyone, I'm Noor. I'm a PhD student with Dr. Rob Patru at University of Maryland. Today I'll be talking about a work which does tree-based differential testing for RNA sequencing, which has been done in joint collaboration with Dr. Michael Love's lab. I know it's been a long session, and hopefully I'll try I make it exciting for you. So we know that a gene can express multiple isoforms because of alternative splicing. And this leads to a problem when we're doing RNA sequencing, is that when we're trying to understand where that fragment, which transcript it came from, that transcript can multimap to multiple isoforms. This is one of the main reasons that we see a lot of uncertainty when we are trying to estimate the abundance of a transcript. So the way we can estimate what we have, we can estimate the uncertainty is by generating additional samples, which we'll call inferential replicates, using sampling of the posterior using approaches like Gibbs sampling. And we have this metric called inferential relative variance, which we compute for each transcript over these inferential replicates. The idea being, again, that higher the value for this particular metric, the more unsure we are about that, the estimate for that transcript. So at the end, any analysis that we do is as good as the quality of the abundance estimate. So we want to make use of this. So how do we make use of this uncertainty? Is like we recently published a paper called Tree Terminus. Idea being that given an RNA-seq experiment, it outputs a transcript tree where the transcripts are arranged in a tree-like structure. Idea being here is that as we go higher up in the tree, the more the certainty increases. Like we are more sure about that combined group compared to the individual transcripts. Just to demonstrate it, if you look in this figure, we have the on the x-axis we have the different Gibbs samples, and then we on the top is a inner node that comprises of two transcripts, and we can clearly see that it has we have much more certainty compared to the individual transcript that it comprises about. And what we want to do is we want to leverage this particular tree-like structure to output a set of differentially expressed nodes rather than transcripts. And these nodes specifically consist of those set of transcripts that exhibit high uncertainty. Just to further, I think, give how the it looks like is like, let's say this is a particular gene, and these are the total set of transcripts that it consists of. And we can clearly see that some of the transcripts have a lot of shared exons. And we arrange these transcripts like this in a tree-like structure. Of course, we don't have to look at the root, but we might be interested in a particular inner node that might be good for the downstream analysis, such as differential analysis. So now we have this tree, but why we can't use the existing tree-based differential testing methods is because they are not directly applicable. Reason being, many aim to improve the sensitivity at the leaf level, but then again, if we are unsure at the abundance estimate, then it's just like we might have just more false positives. Then there are methods that focus on outputting nodes. But then a common approach again is a sort of a top-down approach where you stop going searching for the space itself as soon as you encounter a node that is deemed not significant. Now, in an RNA-seq, we have both up and down related transcripts, and in many cases, this might mean that we might just stop at the root itself, which definitely is not the case. And again, and then as inferential uncertainty, I've been talking about that. None of these directly incorporate that. Given all that, I would now like to introduce a method, BVAR. So again, we want to output nodes, and I'll demonstrate the method using this toy example that I've created. So given a p-value threshold, we can deem a node significant or not. And then we can also assign a sign change for a particular condition that we are interested in. Plus minus sign indicates that I'm not sure about that sign change itself. And then I've also given the uncertainty threshold. So the final set of output node, which is a candidate node, is a subset of the significant nodes. And the significant node should meet this following criteria. If a node has any children that is at least one non-significant child node, or it's a leaf, then only it's a candidate node. That's why we have N1 and N2 and not N18 as candidate. Then all the descendant nodes that display a confident sign change should have the same sign. That's why I have N5 and not N19 as a candidate. And the final condition that we have is that all these child nodes should have a mean inferential relative variance above a certain threshold, and that's why we have N21 and not N17 as a candidate node. So I think the crux is that we are sort of finding nodes. The level is at the node level, which we are sure about the expression. And now I demonstrate it on a real bulk RNA-seq data set, which has been taken from the different muscle tissues of mice. Specifically, we are comparing two groups here, masseter muscle and ADL. So this is a particular gene that, so what you did, when you're doing the analysis, we found that there existed many genes that the differential testing method deemed significant, but none of its underlying transcripts were found to be differentially significantly differentially expressed. But if we arrange them in this particular tree-like structure, we do find an inner node. And if you look at the bottom red two transcripts, you clearly can see that the only difference between them is based on the starting exon. And even the third red transcript on the top, it's also very similar. Only thing is that it also has the retained introns. And it would be very hard for any quantification method to be accurately resolve the reads to such transcripts. So we also plot the inferential replicates. On the left is the transcript that had the lowest p-value but was still not significant. And on the right is that candidate node. Clearly, we can see that the signal has improved by looking at this level. Of course, we do need to run it on a simulated data set. We ran on many. In this particular thing that I would like you to focus on is that we not only are comparing between methods, but also the different features. Like we have gene features, we have transcript groups, and then we also have transcripts. So in a way, when we are doing computing these metrics, these are not directly comparable, I understand, but then we still need a way to understand how the method is performing. If you look on the top, our method on this particular simulation, it has a good sensitivity, a very high TPR, and we are doing good at we're controlling the FDR at different nominal thresholds, barring at 0.05 where we slightly exceed that. But we are definitely doing much better than transcripts. On the other hand, if you look at on the rightmost, which is tree climb bar, which has this condition that if a node is if you get a candidate node, then all of its underlying transcripts are also differentially expressed. In that case, we see while the TPR is high, but so is the FDR has also gone is also inflated. And if we also just look at the nodes, both the TPR and the FDR fall off. So to summarize, we propose a new tree-based approach that incorporates uncertainty, and then we are able to improve the sensitivity and find the candidate transcript through these inner nodes, which would have been missed doing a leaf level analysis, which is by that I mean the transcript level. The code is available on the T-Terminus and BUR. Both are different packages that are available on GitHub, and the manuscript is currently under preparation. Finally, I would like to thank my advisor, Dr. Rob Atru, and Jason, who's another PhD student in the lab, and a collaborator, Mike, and a student, Yufi. And thanks to BioC for the fellowship that has allowed me to travel to this conference. Thank you.
Don't run away. Don't run away. There might be questions for you. We're going to do questions. Yeah. Does anybody in the room have any questions first? Yes.
Is there any concern with like data double dipping, given that you're using the data to cut the tree and then also do the testing?
Yeah, that's a bit.
Get the question.
Okay, so the question is like, is there any concern regarding double dipping, because that same data is being used for tree construction and is also being used for differential testing? This is, I think, a very valid concern we've had since a very long time. But on the simulations that we have seen so far, we did not find as such that we were inflating the FDR. But I've explored the literature for trying to solve this with respect to the particular filter that we have. I don't have a good solution right now. So if anyone has any suggestions, I'm looking forward to it for sure.
Is there anything online?
Another question while we get the next one up, I guess.
Any other questions?
So are you saying you can call sometimes differential nodes that are neither transcripts or genes? Is that true? Sometimes you like don't know exactly which transcripts there are, but that you know that node in the tree is differential?
Yeah.
So that seems like very cool and very accurate, like as most accurate as you can be. But it seems like it might cause a lot of downstream problems for what are you going to do with the data next, and like how to represent it visually and things like that. Have you given thought to those next steps about how to handle these?
I guess like I think it also depends on what level you want to look at. So perhaps if you find a gene, I guess, but then you want to know which transcripts are. This would perhaps help you narrow down the sort of transcripts or look at the different alternative splicing patterns that you're interested in.
Awesome. Thank you. Thanks again. Our next speaker is Dr. Matt McCall from the University of Rochester Medical Center. He's an associate professor of biostatistics and director of the statistics PhD program. His talk is pre-processing and analysis of microRNA-seq data.
All right, thank you. And it's wonderful to be giving a talk to actual living people. Giving virtual talks is pretty miserable, I can attest. So thank you all for being here. I want to start out by talking about what makes an microRNA special. So microRNAs, unlike messenger RNAs, are transcribed in some sort of goofy ways, and there's some post-transcriptional processing that happens. And so the thing that's going to be most crucial for this talk is that we have these sort of two ways that microRNAs can be transcribed. On the left, we have what are called microRNA clusters. So this is a whole group of microRNAs that are transcribed as one initial transcript. And then there's also microRNAs that are in the introns of other genes. And so these sorts of ways of transcription actually cause some weird differences. And so one of the things that happens when we compare microRNAs to messenger RNAs is we have a small number of microRNAs that use up a huge amount of the reads. So on the left-hand side, you can see that for certain cell types, 20 to 30 microRNAs are using up over 90% of the reads in a sample. The other thing that happens, and is sort of related to that, is that if you look at two highly expressed microRNAs, just due to the sort of compositional nature of sequencing, you get this strong negative correlation you see on the top. That's not true for messenger RNAs because there's a lot more of them, the signal's more spread out, so you don't get this sort of induced correlation. So we have people in the lab working on both of those problems. I'm actually not going to talk about those. I'm going to talk about something that we thought would work and only sort of works. And so we got excited. There was a microRNA benchmark data set that came out. And one of the findings of this paper and another paper that came shortly after it was that including UMIs in the microRNA sequencing library prep, this claimed made things a lot better and solved sort of a lot of these issues. So we wanted to see, okay, does this actually work? And so in particular, the UMIs that are used for microRNA sequencing in this benchmark data set and in general, most of the data sets we've seen are based on these sort of random sequences that become before and after your target. So it's generally 4 nucleotides before your target sequence, 4 nucleotides after, and then after. And so we're going to be comparing the two approaches, sort of ignoring UMIs completely and then doing computational deduplication, where we remove things where we see the exact same target and the exact same UMIs. And so when we did this and we looked, here we're plotting, in essence, the difference between our estimated log fold change and the nominal log fold change in this benchmark data set versus the nominal log fold change. On the top, we have the computationally deduplicated data. On the bottom, we have the data where we ignored UMIs. And you can pretty clearly see that the deduplication is introducing this bias. And in particular, if you look at the sign of this, it's attenuating things, right? So we're underestimating the log fold changes in both the positive and negative direction. So we were a little bit stumped on what was going on. And then we remembered this sort of famous statistics problem, which is the birthday problem, right? So this is asking, what's the probability that 2 people have the same birthday in a room full of people? Sort of what number of people are necessary to get to that 50% mark. And so it's somewhat surprisingly, to at least intro stat students, it only takes 23 people. So if we then now start thinking about these UMIs, we have 4 to the 8th, or roughly 65,000 potential UMIs. It only takes 300 of the exact same target molecule to reach that 50% probability that we randomly get the same UMI with the same transcript. So perhaps even more important is the expected number of these collisions, these situations where we randomly get the same UMI and the same transcript. And so it's given by this formula. There's a bunch of approximations. They all give basically the same answer as your n's big enough. And so as you can see, as we go out to the right, 25,000 is well within the number of unique microRNA transcripts that we see in a sample. And we're getting, upwards of 4,000 collisions. So all of those calculations are assuming that UMI transcript pairing is random. And so when we looked in aggregate over the different pools and the different labs and this thing, there's some deviation from sort of a 25, 25, 25, 25 usage, but it's not too bad. However, if we look at the UMI usage that we see for a specific microRNA, we see that it's not random. In particular, for this specific microRNA that I'm showing, that 5th position is almost always a G. And so this is suggesting that there's we think it's a ligation bias. This is the G that immediately follows this target sequence. And so there's some aspect of that that's causing the UMIs to not bind randomly or not be incorporated randomly to the target sequence. The other thing that is particularly problematic about microRNAs, right, is that they're short, right? And so there's no sort of random fragmentation happening that you might have with a long mRNA transcript, where there's additional variation in that UMI sequence pairing due to the fact that you're getting different target sequences based on different regions of the messenger RNA. Here, you're sequencing the entire microRNA transcript every time, right? They're only about 21, 22 nucleotides in length. So one of the things that was interesting in this benchmark data set is they actually included a few transcripts that were longer. So up at the top of the figure are things that are 90 nucleotides long, then 75, and so on down. And we see that for those things where they're a bit longer, so we're not always sequencing the entire target molecule, we get less of this bias in use. So the other thing is that microRNAs, right, as we talked about in one of the early slides, are often very highly expressed. And so actually the bias seems to be driven a lot by the most highly expressed microRNA. So even if you have a microRNA that, is short, this sort of collision probability isn't going to be that high unless the microRNA itself is quite highly expressed. So we actually looked at this. Here in the bottom, we have this sort of volcano plot-ish thing where we have the bias on the x-axis. We have the square root of the max of the count in either condition A or condition B. And we actually are using this relationship here. We model this relationship and use that to adjust our full chain estimates. So does this actually work? Well, sort of. So we do better than the computational deduplication for sure. You can see how sort of bad the MSEs are for the very highly expressed things, both on the high and low ends in terms of full change. But we don't quite get as good performance as just using the counts and ignoring the lines. So that's my very brief talk about this project. If you have ideas, I'd love to hear them. We're sort of at the point where we're debating whether to just like write this up and say, hey, there's this problem, we can sort of solve it, but not really, and leave it at that, or, you know, hopefully in the next few months get something better to work on, or to work better for this. So most of this work was actually done by a grad student, Samuel Young. The rest of the people are some talented postdocs, a research assistant professor, a master's level statistician, and another poor PhD student who's trying to sort of figure out something else to do. This is in collaboration with Lushka's lab. And yeah, so we have some fun at GMS. So thank you all.
So over here again.
So given that the relationship between expected collision, like number of transcripts and expected collisions is well known, could you derive that same equation for the non-uniform UMI distributions and use that rather than having to model it empirically?
Yeah, so that's a good idea. There's actually one paper that came out, I think, 5 years ago or so.
Is this accidentally NP hard or something?
No, it's like DROP-S, I think. It was for single cell mRNA sequencing data. But they were trying to actually sort of come up with a way of estimating sort of these probabilities semi-empirically, but also sort of combining some of these equations. We tried it out, it didn't really work for like RNA sequencing data. That's not to say that there isn't something there. We've definitely tried, you know, correcting based on sort of the uniform binding assumptions. We've tried to sort of think about, can we actually model the sort of non-random binding? That didn't really work too well, but that's not to say that it couldn't work.
Anybody else? You blew them all away. You explained it so well.
Thank you so much.
All right, our third speaker is Dr. Sean Madden, whose LinkedIn, university profile, and Twitter all say different things about where he's working. But I'm going to take a guess and say he's a computational biologist, a postdoc at Johns Hopkins, and his talk is going to be LUT, a new framework for bulk transcriptomics deconvolution experiments.
Thank you. It's Maiden, actually, not to be confused with the late great John Madden. So yeah, it's great to be here in person, and I'd like to talk about LUT, which is an R package that I've been developing. It's a new framework for bulk transcriptomics deconvolution experiments, and I'm presenting on behalf of myself and my amazing postdoctoral mentor, Stephanie Hicks. And so I'd like to start off with a brief overview of deconvolution. So this is the problem of quantifying cell types from cell mixtures. Here we assume non-negative expression, and we also have a number of terms and representations, which I'll explain. So the term Y is the bulk expression signal mixture, or the mixture of cell types we either predict or which we take from a reference. And we have the pseudo bulk representation of that Y term, which is as the product of the Z term, the cell reference expression. So that's like this atlas with marker genes by cell type specific identities or labels. And then in that product term, you have known cell type proportions for pseudobulk, which has the same K-length vector of cell type proportions. And I want to emphasize that the kind of in this setting, we're taking a reference, an assumed reference that's reliable and that we can use on cell type specific expression signatures. And then the Y term and the Z term both share this axis of the same G marker genes on whose expression we base our findings. And this diagram is meant to be a helpful depiction of some of those terms and how we might visualize those in bar plots and heat maps. So there's not enough time to talk about all of kind of the challenges and opportunities in the recent literature in bulk transcriptomics deconvolution. So I wanted to highlight this preprint as a citable unit and as something that we published this year with our amazing collaborators at Lieber Institute for Brain Development. I encourage you to read that if anything in this lecture strikes your fancy or is of interest. But one of the key takeaways from that effort is that many recent efforts to perform bulk transcriptomics deconvolution involve multiple platforms, assays, and orthogonal assays in particular. And so there's a variety of different data types. Among them are simulation-based data sets and result sets, as well as real data-based result sets. And then these need to be basically organized. And even when considering a specific tissue or a specific anatomic region within an organ or tissue, there's this need to, an unaddressed need and also an emerging issue that I see for organizing multiple assays along common axes or indices. And so one example of a way that we could improve conditions for data sharing in this setting is to use standard and high quality data containers available through bioconductors such as multi-assay experiment. And in addition, when reviewing recent literature, I concluded that there's a lot of kind of data forward bias correction and data forward processing and algorithm development. So one example here is with correcting for systematic bias in bulk assays to match the bias that we observe in a single cell or single nucleus reference data set. And there's more to unpack there, but this is an exciting area of continued research and not something that I want to discourage by any means by saying that we can improve more. But I also want to say that there's a certain threshold or barrier to access for understanding the way that new algorithms work. And this includes common shared tasks for these algorithms to ensure that the data that we're passing to them meet certain quality standards, and also that they're processed in uniform ways, and also needing to map standard terms that are shared across the different algorithms. And so this is a figure taken from that preprint, and we have in the panel an example of summarizing from single cell level expression to a cell reference atlas, or that term Z in that standard equation as shown. B is the panel depicted earlier, and then that S term is a cell size normalization factor, and then that is unpacked in panel C here, where we demonstrate an output in which we have the true or reference that we take for a given cell type in a sample on the x-axis, and then the predicted cell type proportions on the y-axis. And we can see the results in the scaled are more closely aligned with true compared to predicted than in the unscaled case on the bottom. And so why use LUT? LUT takes its name from deconvolute. This is a starting point for complex experiments, specifically focusing on the deconvolution setting. LUT is a framework, so it actually provides a function that enables type marker access with a generic function, as well as deconvolution with a generic function. And it takes strong inspiration from a prior effort called Bluster, which provides a similar generic access to multiple different clustering algorithms. So this flowchart is meant to depict kind of some of the tasks that LUT is trying to take care of. And one of these is to manage multiple different input data container types or data structure types, and to pass those to common terms in the type marker generic, which are then passed to common terms in the deconvolution generic. Now, this is not intended to be like an obscuring layer or a black box of any kind. I actually want to encourage like development on each of the different layers depicted here. but it is meant to say, well, there are certain common terms that we can agree on when we go to develop a new method or algorithm. And one of those things that we can agree on is when terms are synonymous with one another. And so to unpack that a bit further, LOOT is meant to save lines of code by saying, okay, if we want to perform a benchmark, if we want to compare my new method, or if we want to compare an old method to a new method, then we should have support basically for performing those types of experiments out of the box. And so one way to do this is that I proposed and that we got to run with LOOT, again, basing this on prior work, was to define a series of class structures, and that's depicted in this hierarchy at the top. And in that organization, you have the different terminal nodes or the different algorithms, and then the hierarchical class structure is the management for different algorithm types. And this table is depicting like what makes it all work. So each row in the table is a different algorithm, and then each column is metadata about that algorithm. Some of these are newer, such as music, epic, and DNCon RNA-seq. Some are older, such as NNLS. And then the terms Y and Z, as defined earlier, are mapped to different arguments in the code, but they also appear in the math that's represented in this publications as well. So if you want to learn more about different algorithms that could potentially work in this kind of approach, I'm beginning to track them in something called Awesome Deconvolution that you can check out now. And then as I'm developing LOOT, I'm also conducting new research with our awesome collaborators at Lieber Institute. And this is in human dorsolateral prefrontal cortex, which is depicted in the panel at top left. And this is a region of the human brain that where there's growing amounts of reference data sets that are being collected, as well as matched orthogonal assay types that are being collected from neurotypical donors. And then we're processing these on 10x Chromium platform, and that workflow is depicted at bottom left. And then to begin a deconvolution experiment, we need cell type labels from somewhere in terms of real data experiment rather than pseudobulk. And so from cluster analysis, we have assigned a number of cell type labels as shown here. These include both neuronal cell type subpopulations as well as glial cell type subpopulations, and the bar plot is depicting their absolute nuclei abundance. So this is a single nucleus assay rather than single cell. And then we could take the expression data from these real data sets and base pseudobulks on that real expression data to sort of anchor them in the real world, so to speak. And one example of a pseudobulk simulation that could be useful here is to simulate situations where cell types differ. They're, one other way to say this is they're highly heterogeneous. In the case of human brain, especially, neurons are much larger than glial cells. So if we take real expression data and then take the cell labels and say, look at what I would call a K2 case is depicted at the bottom right. We have, again, the true cell composition by the estimated cell composition, and that reference line representing 0 error. And then the distance from that line representing how far off the deconvolution algorithm is. If we throw an algorithm like NNLS at this, we can depict the principle that if we perform a normalization on the distinct cell sizes in a heterogeneous tissue, that we can remove some of that error. Some other things to note are the sides of the large cell type, which is systematically over-predicted, and then the smaller cell type is systematically under-predicted. There's also support for another not an algorithm, but a workflow. So this is meant for benchmarking deconvolution algorithms from that are supported in R, and LOOT allows generation of a flat table that's basically compatible with this new workflow that's available on deconvolution, and you can run Nextflow and deconvolution benchmarks right now. So in conclusion, LOOT is addressing what I find to be an unmet need for management of complex data types, multi-assay, multi-platform experiments in the deconvolution problem space specifically. And it helps to normalize across these types and experiments. And the goal is to lower the barrier to access and activation barrier for performing deconvolution experiments in transcriptomics. So it's under active development. We appreciate your feedback. And then I'd like to acknowledge and thank my amazing mentor again, and collaborators and colleagues at Johns Hopkins School of Public Health and Lieber Institute for Brain Development. Thank you.
We have time for a quick one, so I'm going to steal it myself because it's my privilege. There's definitely, a use case for these kind of tools, and I think it adds robustness to existing data sets. We can go back to our old data sets and apply these kind of things. So I'm really glad you're working on these problems. One problem with some of the other tools is when they aren't confident in their deconvolution, when the confidence intervals are too low, they still try to assign things. How do you address that sort of problem so that you're not chasing ghosts?
Yeah, that's a great question. Actually, confidence intervals for deconvolution outputs is a great application for something like LUT, which I would say the goal is to be able to systematically QC like systematically QC error testing and also error in different settings. And so a confidence interval would be a great application for what is essentially right now a glorified wrapper, but could ultimately be a new way to form like an empirical query or empirical experiment.
Thank you so much. Our fourth speaker is Dr. Oh man, I'm terrible with names. Let's go with I can't enunciate. It's like a name thing, guys. It's not like it's a hard name. Jean-Philippe Fortin. Hopefully I've not butchered that. Senior Principal Scientist at Genentech. I'm just going to bring up your slides real quick, but you can jump in right when they're up.
Go by JP.
JP, thanks JP. I'm so sorry. Like you should, yeah, I'm really bad. I used to be a camp counselor and it was really bad for my kids.
Hi everyone, I'm JP. Super excited to talk about the CRISPR-verse. All right, so a quick introduction to CRISPR. CRISPR-Cas9 is a very cool system. There's two components. The Cas9, its job is to cut the DNA, but then you can guide it to a specific location in the genome, and that's what the guide RNA is doing. And we can design this piece of 20 nucleotides to a target sequence in the genome. And when the two binds together, it's gonna cut at the target location. It's going to introduce a double-stranded break, and the cell is going to repair itself. And you hope that it's going to be an imperfect repair, and that's going to generate a knockout. And so the whole job of designing the guide RNAs is to find this perfect sequence that is complementary to the target location, but you also want it to be not too similar to other locations of the genome because the nuclease is also tolerant to mismatches, so you don't want to create off targets. So when I started at Genentech, that was 6 years ago, it was super simple. We were doing one type of screens, but since then we've been expanding the toolbox of what we do with CRISPR. We do a lot of crop-seq, perturb-seq, also optical pole screening, which is combining imaging and CRISPR perturbations. So my job as leading the bioinformatics for CRISPR at Genentech is to design those experiments with a scientist and also to do the analysis of those experiments. So since I started, we developed an end-to-end pipeline to do the design, analysis, and the visualization. And we created this whole ecosystem of R packages and Shiny apps. So this is what the current state of the affairs at Genentech. But today, I'm going to talk about one part that we were able to open source and publish on, which is the CRISPR-verse. So that's the very first part, designing the guide RNAs. And as I said before, it was super simple at the beginning, CRISPR-Cas9, but we've been extending the CRISPR into many directions. So we do inhibition, activation of genes, base editing, and we're also testing different nucleases, also some RNA targeting nuclease like the Cas13D. So it's become quite complex to design those guide RNAs. And a couple of years ago, there was a pandemic, so we kind of restructured the whole ecosystem to be more modular and also publishable. And at the same time, we looked at all the existing tools that were publicly available and that we could use at Genentech. And we realized that most of the tools just don't have the functionalities that we needed for the experiments at Genentech. And you can see here that there's a lot of white spaces, and there's no particular tool that does everything we wanted to do. So the rows are the different functionalities that we needed to do for the experiments. So that kind of motivated the open sourcing and being able to publish on it. So the way we designed the ecosystem, we made it modular and reusable components. So in the box in the middle are the different components that are now in Bioconductor. And you can see that they're not specific to our ecosystem. Everybody can reuse them and they can build on top of them. So CRISPR bowtie, CRISPR BWA actually have wrapper for bowtie and BWA. You can use them for anything else. And that work was not done alone. So that was done with Luke, Aaron, Piru was here, and Nor, who just presented, was a summer intern Genentech with me this year. And also a lot of the Bioconductor reviewers that I want to thank for their help reviewing the packages and making them better. So just a quick overview of how the CRISPR verse is working. We start with the genomic region, represented as the genomic ranges. And we kind of build a guide set object, which is an extension of genomic ranges. And then we add those very complex annotations. So the beauty of the genomic ranges, the metadata columns can be list columns, so we can store complex objects in the columns. And one thing that might be of interest of the people today is the CRISPR score package, which was by far the most challenging package. I will tell you in a minute why. All right, so the great thing so Aaron is actually, Aaron is living a block away from me. We see each other's window, which has pros and cons. But one of the pro, at least before pandemic, is we used to take the bus every day together to go to work. And I can just bug Aaron about my problems every day. And one was that most of the algorithms to predict the efficiency of the guide RNAs are done in Python. And they've been done over the years, and they are not maintained by the academic labs. And some of them are in Python 2, in Python 3. So I was asking Aaron, can I run all of those in 1 R session? He's like, yeah, I'm actually building Basilisk. So we spent many months cleaning up each of the Conda environment for Windows, Unix, Mac, which is very tedious, as you can imagine. But once those Conda environments are frozen and we basically have the recipes in the R package to build them, it runs pretty smoothly. And you can run basically those, we have, I think, 9 Python environments that we can run in 1 R session now. And at the end of the day, the users see this very nice table of scores, but under the hood, it's been using 9 Conda environments, which is pretty cool. At the end of the day, when we have all those rich annotations, the goal is to pick the best guide RNAs. So we design a ranking algorithm for different species, different applications like CRISPR-A, CRISPR-I. And then I don't have the time to go over this, but we did a lot of benchmarking using public data sets and using all of the public tools. And we were doing really well. So CRISPR design is kind of the core package of the CRISPR-verse. So we were very happy to see that the ranking and using the functional annotations, we were able to get very good guide RNAs. And we have a CRISPR, a GitHub organization with all of the packages, with very extensive tutorials that were requested by the reviewers of the paper. And then everything is in Bioconductor, and we were able to publish our work in last November. I want to thank everybody, our collaborators, but I also want to thank Michael. Michael is the director of our department, who is a proponent of open sourcing and publishing, which was great for our work. I'm going to end on this. So there's an open position on in my group to work on genomics workflows. So come talk to me. I would be at the poster session after this. Yeah, thank you.
All right. Any questions from the room?
Do certain of these algorithms predict better in certain systems? And do you, like, if that's so, do you know why?
The question was, do these algorithms work better in different systems or have a bias?
Yeah, some of them were trained in zebrafish, for instance, with a different promoter. So there's very promoter-specific algorithms. There's also modality-specific algorithms, so CRISPR-A, CRISPR-I, and right now, NOR is actually working on a project this summer to do transfer learning to be able to use a previous data set, to generalize the algorithm across modalities. Yeah, that's a good question.
All right. Thank you again. Awesome. All right, our last speaker for this session is PhD student from Cold Spring Harbor, Renee Garcia Flores. Her talk is non-invasive, low-cost RNA sequencing enhances discovery of potential of transcriptome studies. I might have added extra oven there, sorry.
Hi, I actually haven't started my PhD, so. But yeah, this is the project I'm going to present, but I work in this project in the summer, from the summer of 2021 to 2022. And a little disclaimer, this is not the whole project. I'm gonna focus on the parts I mainly help with. I was an undergrad at that time, but yeah. So in the last decade, when trying to answer questions of biological importance and disease-related, GWAS studies have been used to correlate regions to particular phenotypes. However, because of the fact that we are only taking into account genomic data, it's not possible to know the functional consequences of these variants or understand their role in a given tissue during biological processes. Also, in addition to this, previous studies have shown that variants in GWAS catalogs show an enrichment for ccQTLs and ccSQTLs, elucidating regulatory mechanisms of the variants detected by GWAS that can only be correctly identified and characterized when including transcriptomic data, making it important to perform more studies, including both data types. However, obtaining transcriptomic data has certain limitations. One of these limitations is the invasiveness and limited disease relevance of tissue types that can be collected. For example, whole blood, which is one of the most used tissues to perform transcriptomic studies, has shown to have expression patterns that segregated from other tissue types, which can cause an important bias in the detection of variants. Similarly, postmortem tissues, despite we no longer have limitations related to invasiveness, other problems come around related to the fact that now we have to deal with things like degradation, and also that the sample size is very limited in this kind of source. Another alternative is to obtain tissue from surgical extractions, which, although is a good option, represents a stress for the patient. It also has limited sample size and, importantly, adds an expense on top of the already high cost of sample collection and library preparation, which is the second limitation when obtaining transcriptomic data. Given this, with the main goal of finding other alternatives that allow us to generate data of good quality for functional analysis, while lowering the invasiveness during the sampling and also lowering the cost in the library preparation and sequencing, Molly, the PhD student I work with, developed a project. For this project, she took samples of 4 tissues that do not require surgical extraction, which are saliva, hair follicles, and buccal and urine cells. The samples were collected from 19 donors in 4 different time points, separated by 2 to 10 days. And to lower the cost, Molly developed an in-home library preparation that I'm going to be referring to as LowSeq. She used this LowSeq prep in all the samples, and also used Illumina TrueSeq and SmartSeq for control. After sequencing and mapping, Molly noticed as we can see in here, that the mapping rate is different between tissues, being hair and HEG, which is the control, the samples with the best and most consistent mapping rate, and saliva and buccal, especially saliva, the most inconsistent and with the lowest mapping rate. This made us wonder, well, at this point, I joined the project. I wasn't in the sampling. But yeah, this made us wonder, why are these sequences not mapping? Could it belong to microbiome? Maybe lab contamination, or maybe could this be happening because of technical factors or intergenic repeats? We didn't know, and answering these questions was really important, since we are using a novel library prep, and we wanted to check if that wasn't adding some kind of bias. And also, we wanted to explore all the possible applications of this methodology. So to answer these questions, we first remap the unmapped reads to microbial genomes. This process remap only a small fraction of the unmapped reads, but when we look at the top 0.5% of most abundant species, we observe a distinct microbial signature across the non-invasive tissues that support the previous known microbiota of oral cavity, human skin, and genitourinary tract. In addition, we see high correlation in estimated species abundances across technical replicates, suggesting that despite the limitations of our library preparation approach, we are capturing real replicable biological signal. Altogether, this finding supports non-invasive samples may bear biological utility in follow-up microbiome studies using microbiome-specific orthogonal RNA library preparations. Next, to investigate the biological similarity of non-invasive tissue to known invasive sample types, we compared the gene expression and EQTL discovery to GTEx. First, we projected the non-invasive samples into the GTEx PCA space to observe global patterns of gene expression similarity. Hair clusters closely projected with esophageal mucosa and skin, and saliva is proximal to spleen, blood, and EBVs, and buccal and urine are intermediaries between these groups. Then, to further explore how non-invasive samples capture genetic regulation variants discovered in postmortem tissues, we asked the replication of GTEx EQTLs in buccal, hair, and urine samples. Here in the graph, I'm only showing the results when using hair, but we can see, okay, so the dots show the P1 calculated by selecting significant GTEx gene variant pairs from our samples. And in the violent plots, we're showing the null P1 distributions generated by randomly selecting gene variants. Gene variant pairs. These, along with other analyses that I'm not showing, suggest gene expression regulatory mechanisms present in invasive tissue may be captured non-invasively. And finally, to explore whether RNA sequencing from non-invasive tissue may be used for differential expression analysis, we tested for sex-based differential expression and the biological role of the discoveries. The hair results, which you can see in the right, show significant enrichment for checkpoint pathways in female, which are central to regulation the cell cycle and proliferation. And in males, although no significant WNT signaling has previously, which was the top hit in males, has previously been reported to play a key role in hair loss prevention. And in urine, notably estrogen response was really enriched in females. This analysis demonstrates that the potential for non-invasive samples to elucidate underlying biology and a variety of potential contexts and assays. And as conclusion and perspectives, we were able to characterize many of the technical and biological features of or for possible non-invasive samples. We conclude that urine and hair follicles were the most robust tissues, but we think that our findings will need to be validated using larger, more diverse and clinical cohorts. And acknowledgement, Molly is the MD PhD student I work with, and Tuli Lapalainen, the PI. So the two of them are such a great tutors, and I have a great time at the lab. And also, I would like to thank to the collaborators, and the Lapalainen Lab, the New York Genome Center, Columbia University, and my undergrad program. And if you want to see more of this project, here's the QR code for the preprint, the QR code for these slides, and you can find me in GitHub and Twitter.
Perfect. Right on time too. Any questions from the room to start? Yes.
If you were able to access matched pairs of these tissues plus the invasive tissues that are hard to access, do you think you could develop a model to impute the expression in the hard to access tissues based on the expression in the easy type?
Okay. Could you repeat your question?
So if you could access these tissues plus also the hard to access tissues from the same people at the same time, do you think that you could develop a model using the expression from these tissues to impute the expression in the hard to access tissues?
Okay. So the question is, if I had access to these two types of tissues, the non-invasive and invasive tissues, if I could create a model to impute the data from the invasive tissue to the non-invasive. I will say, I will say yes, given the results from the GTEx and also other expression analysis we made, and I'm not showing. But I mean, I think the project needs more analysis related to it. For example, hair, it's a good sample for skin, but maybe if I'm taking buccal samples, to which other tissue will it correlate? And yeah, I will say we need more non-invasive tissues to use, and also getting the invasive tissues would might be hard. But yeah.
Question for the pi 1 estimates. So did you look at, if you split that by promoter and distal regulatory variants and their effects, like is a lot of the sharing at the promoter?
Okay, I'm not able to answer that question. I'm sure it's in the paper, but yeah, sorry.
Yeah.
Thank you.
That concludes the RNA Short Talk session. Thank you, everyone, for joining.